Thanks, Dusty. I appreciate the opportunity, and I'm really rethinking how I'm going to do this since we've had two really good talks this morning about how best to present stuff. So we'll see whether I can actually make that work. So I think maybe I'll start here. As Dusty says, I wear a lot of hats. I got my PhD in 1994 in immunology from the University of Toronto, working on ruminant immunology. Since that time, I've basically focused on a combination of ruminants, and frankly, I've branched out to comparative immunology, where I've worked on just about any species you can name through collaborations or otherwise. After I got my degree in Toronto, I moved for about 5 years to work in Switzerland at Hoffmann La Roche in a privately funded research institute as the head of the ruminant immunology lab. When they closed that down, I decided it was time to find another job, so we moved to Boston, where I spent a year playing at being a faculty member before we decided that Boston wasn't the best place for us. Moved to South Dakota in 2001, been there ever since on faculty at the South Dakota State University, affiliated with the Veterinary Diagnostic Lab in the Department of Veterinary and Biological Sciences. For the first decade, I guess, of time I was there, I spent most of my time working on mad cow disease. But starting in about 2009, I started working with Kansas State University in the Center for Emerging Zoonoses and Animal Disease, which is foreign animal disease based, as one of their principal investigators working on the Rift Valley fever vaccine. And then, as Dusty mentioned, in 2011, I decided I'd had enough free time, so we started up a biotechnology company to start commercializing some of the research coming out of the university and out of CZAD, and one of our first targets was actually the Rift Valley fever vaccine, which we are currently in process of licensing. We are now a fully licensed company producing autogenous vaccines. We are awaiting our first product license, and that's part of what I'm going to talk about today. So given that I'm supposed to open with a story, and this was not part of the original one, so we'll see how this goes. Many of you are probably aware. So we were a young fledgling vaccine company working on Rift Valley fever because of all of the commercial relevance of that. We'll talk about that in a bit. And then 2013 rolled around, and a little virus called PED came out in the middle of the heartland in the United States. And PED was kind of a wake-up call for a lot of people in a lot of ways, but one of the things that is directly relevant to what I'm going to talk about today is how it was a wake-up call for the regulatory agencies. Here we were struck with a virus with devastating consequences for the animal industry, and there was no way to produce a vaccine that could protect against that disease, despite the fact that it was quite homologous to TGE, so we pretty much knew the targets that we had to pursue. But following the normal regulatory pathway would take, at best, 16 months, 24 months, something in that ballpark. And so, to give the CVB credit and the regulatory agencies credit, they went back, they worked with a small startup company to come up with new regulations that made sense. It was a little bit of a shock to me as a university professor to find out that the majority of the regulations, or the regulations we were working on in 2014, had last been revised in 1983. In 1983, I was a high school student. Nobody had actually started cloning DNA. PCR was a glint in Carrie Mullins' eyes. And so it was time for them to update these regs. And to their credit, they did. And so that's the basis for what I'm going to talk about today. It's the basis for the new vaccines we're developing at Medgene. And it's the basis for a proposal that we're looking at as far as looking at a reasonable approach to mediate foreign animal diseases. And what it basically does is it allows us to use the knowledge that we have gained over the last 3 decades on the use of molecular biology or recombinant vaccines to generate safe, effective targets to vaccinate against diseases, and to develop those vaccines in a fraction of the time and for a fraction of the cost that it would take to develop a traditional vaccine. So, the last thing I should say, which I should have said at the beginning, so I am a university professor at South Dakota State. I am a business founder and owner of Medgene Labs, but I am speaking today from my experience in my past as me personally. Should not be assumed to be representing South Dakota State University in anything I say. My business owners would tell me not to assume that I'm representing them, and my wife would most definitely not want me to say I was representing her. So, some of this I'm sure you're more aware of than I am, but it's relevant to our business model. As I said, we are currently pursuing licensing of a foreign animal disease vaccine, Rift Valley fever, disease of ruminants that is not currently in the U.S. It's mosquito-borne like West Nile virus. It's zoonotic, so it's a threat to the human health population. And so we've had to experience a little bit about what we're planning on in terms of a marketing strategy for this vaccine. And so that's directly related to the federal guidelines. And so if you go online, as I did, and some of you, including some of them in the room, probably are more well aware of what this involves than I am, you'll basically see that a foreign animal disease plan has three components to it. Stamping out. Find the thing, drop a bomb on it, get rid of it before it spreads anywhere else. So containment, number one. If that containment fails, the next step is to limit the spread. You can do a ring vaccination, or you can try to control animal movement by shutting it down. Whatever it takes to confine that infection to that regional area so it doesn't spread, making the situation worse. And finally, if it gets even beyond that, you want to be able to resume business. I've seen this in the resolutions you were just talking about. You recognize the need to have the ability to continue to move animals, to continue trade in the face of one of these outbreaks. And so there are a number of characteristics associated with this. One is vaccine, so you can defend against the disease. Two is diagnostics, so you have to be able to detect the agent. You have to be able to detect it quickly, and you have to be able to detect it regionally. You have to be able to detect whether animals have been infected or not, so you know whether it's safe to move them. And finally, if you're going to use a vaccine optimally, not only do you want it to work, but you want to be able to tell the difference between a vaccinated animal and one that's serologically positive because it got infected. So how do you go about doing this? Well, current plans for vaccine banks responses at the federal level involve things like looking at offshore production of vaccines, a vaccine bank. So why wouldn't we want onshore production of vaccines for foreign animal disease? Well, as a company owner, I can tell you right now that Dusty wouldn't be too happy if I said we were going to start making foreign animal vaccines in Burking, South Dakota, so we were going to bring in 23 strains of FMD, Rift Valley fever, African swine fever, classical swine fever, and start growing that. So nobody wants to touch the bug. Secondly, it's expensive. So there's an issue in getting companies to pick up and travel the final distance to get a vaccine through licensure for use. And we'll talk about that a little bit further on. And so one of the ideas that has been put forward, particularly for FMD, is an offshore bank. So we'll have somebody in a different country produce this for us, and if we ever need it, then they can send it to us. Well, I can tell you as a vaccine company, somebody who's gone through the regulatory agencies, that what you're basically doing is saying, okay, we've developed a regulatory agency to make sure that everything you're doing falls into the right category so that they're safe, that they work, that there's no danger of them making disease worse. But in the case of a foreign animal disease, we're just going to throw that book out the window because we don't have oversight of these foreign companies. We don't have control of their production streams. We don't know if they've gone through inactivation kinetics and inactivation curves to ensure that when we buy that FMD vaccine, it doesn't have live virus in there. I'm not saying that would happen, but that's a possibility. Also, there's no guarantee it will be effective. There are, 23, 26 strains of FMD floating around out there. What if we get strain number 27 or something that's mutated on the way over here? Now we've got an entire vaccine bank that doesn't deliver what we actually need. So those are concerns. The other approach, which is the one that we've traditionally used up till now, is we have researchers working to develop efficacious vaccines that can then be handed off to private industry. And private industry can then take those through the licensing pathway and develop them for use in the vaccine bank. And part of what I'm going to tell you about today is why that is somewhat overly hopeful. So this is kind of where we're at. I'm not sure if everybody got to see Apollo 13 the 15 million times that I did because of my daughter's love of this movie when she was a child. But the classic scene is Ed Harris playing Gene Kranz here has just found out that, in fact, the Apollo 13, for which they had planned for every possible outcome that could possibly go wrong on the space missions, Apollo 13 suffered a catastrophic failure, and they could only get them from the Earth to the Moon and back this far before all the astronauts were going to die. That last little bit, they couldn't make it. And so his job was to figure out how they could make it that last little bit. And that's kind of the analogy that I want to talk about today. Up until now, our approach as researchers and academics, I can tell you for a fact, and has been to develop vaccines that we can then put in a small number of animals and show that they work. And this distance here, this is the part that we've been relying on industry to do. And the problem is that for industry, this is not something anybody wants to get into. And so what we're going to be talking about, or what I'm going to be focusing on today, is a plan that would allow us to use those new regulations that I talked about at the beginning that were brought in response to PED to develop a responsible, cost-effective way to have an insurance policy against something that we hope will never happen but could. And so the basic issue with us getting that last little bit from the moon to the earth, from a commercial standpoint, is cost and benefit analysis, because private companies are driven by cost and benefit analysis. And I have found this out the hard way by developing a Rift Valley fever vaccine. Luckily, my board, my investors, see the commercial relevance of the other things that we're doing, because if it was all based on Rift, I would be in deep trouble. So what I've been told by my colleagues that we brought in is the industry standard to put a vaccine on the market for the animal industry is they expect about $1 million in revenue for each vaccine that they get licensed. Currently, there are 15 agents on the National Veterinary Stockpile list, ignoring sub-strains and things like that. So assuming that we had universal vaccines for each of the 15 agents, that would be a $15 million per year by the stockpile to be a consumer that would interest industry in doing this in order to generate an effective product. Multiple serotypes exist for many of these agents. Right? FMD, what is it? 23 is the number I've seen, I think I've been throwing around. To date, we don't have universal vaccines against these things. Everybody would love to see one. When I was a first-year immunology student, we talked about universal influenza vaccines. I'm now not a first-year immunology student, and we still don't have a universal influenza vaccine. It's a laudable goal. Is it going to happen in the near future? So, FMD alone, we'd be expecting about $23 million in revenue per year for a company to have that as a licensed product in the stockpile. So some threats are not even included on the FAD list. These are the ones that are the foreign animal disease. What about blue tongue 8? It's not on the list. We have PED was never on the list in the first place. There are other things that are coming that we should be aware of that could be a problem that are not on the list. In the Farm Bill, and this is, I realize the speaker coming after me is going to be much more detailed, but from what I've been able to read at that, and I'm not very good at it, it's about $5 million a year allocation. So we're a little short between that $5 million and about the $50 million a year that you would need to do this as a licensed vaccine bank. So let's talk a little bit about these platform vaccines. So what they did as a result of PED is they realized that there are ways to make vaccines against the specific protective targets. For influenza, it's the hemagglutinin molecule, H3. For PED, it's the spike protein. We know this. There's 20 or 30 years of research to tell us that this is what it is. And so you can either get that protein by growing up the bug, killing it, using the whole bug and vaccinating, which is the traditional autogenous kill route, or you can use a different platform-based process to put in the DNA so that you only make the target of interest. And if you only make that target of interest, then you have what's called a production platform. And what the USDA did is they said that if you have a production platform where it meets certain criterion, it's killed, it's safe, the only thing you change is that DNA sequence you put in there, and you get a fully licensed product on the back of that platform. So you have one product that you run through the entire rigmarole for CVB licensing. We will allow you to then prove that that's safe in the field, and then you can sell this as a platform product by hot swapping out the gene of interest and putting in new genes of interest. At a practical level, that means that that first license is just as onerous as any other licensed product for the CVB. But once you have that license, you can then use that same platform to produce new targets. And those targets can be for pretty much anything. And as a result of that, your costs drop. Your time to produce a new vaccine drops to as little as a couple of months or even less. And so you can then respond to emerging strains, emerging viruses, emerging infections. So the comparison I use is that prescription platform products are kind of like pills of penicillin. So if you want penicillin, I did the math on this, so I'm very pleased with it. You can take 2 big cups of mold and eat that, and you'll get about 500 mgs worth of penicillin out of eating that mold. Basically, this is autogenous. We take the entire organism, we grow it, we kill it, we slap it in a bottle, and out the door it goes, and we assume that the component that you want an immune response against is going to be in there. Prescription means, no, we're just going to produce the penicillin. We're going to make a recombinant bacteria that produces nothing but penicillin. We're going to purify that penicillin, put it in a product, and that's how you're going to get your 500 mgs. That's the prescription vaccine. So at a platform level, this is kind of what it looks like. So if you have a virus, and you know that the target protein is this one on the outside, and if you immunize against that target protein, you protect against the virus. For a lot of viruses, there's not a lot of mystery on this. You can either grow up that virus, kill it, and put it in a bottle and have an autogenous, or you can isolate just this protein, produce a system that produces that protein for you in cell culture at the bench, package it up, put it into a bottle, and this will have the same protective efficacy as this. That's a platform. And what makes it special is once you've done this once, if that protein changes like it does with swine influenza or human influenza or anything else, you can go back, you can change the sequence to match this new protein, stick that in a bottle. Now you have the second strain. Or do it again if it changes somewhere else. Third strain. There are currently licensed vaccine products in the human market that do this. There's a vaccine called Flublok for influenza that does this. The other trick about prescription is this, for those of you in the room. It's called prescription for a reason. It has to be prescribed by a licensed veterinarian. And so this is the basis that Medgene's business model is pursuing, is we are pursuing fully licensed products in the commercial realm, influenza, and when completed, we will be able to deliver proteins against highly changing organisms or highly changing viruses that are tailored to the outbreak on that farm or into that region that are prescribed by the veterinarian. The control is similar to autogenous. The efficacy is proven through the licensed pathway, and this is a fully licensed approach. So that's great for commercial markets. But you know what? This is entirely applicable to foreign animal diseases too. So the basic model system then, one more, is similar to the concept of the Keurig machine. And I'm probably going to get sued one of these days for saying this because I never actually asked for permission. But what you license is the machine, the one we have in our rooms right now, right? You go through a license process where you have the machine, you have your basic coffee pod for regular coffee, and you run that through the full licensing procedure associated with the USDA. But when you're done, you have a license for this system. So if now you want to produce French vanilla or Irish cream, then all you do is you substitute that out, you put it in, you notify the USDA you're doing it, but there is no regulatory process, and you can produce those different things. And the same thing is true for our vaccines. So what that means is that once you have a license for this machine, you can produce swine influenza vaccine, or you can produce PCV, which is on the same backbone, or you can produce Rift Valley fever vaccine, or classical swine fever vaccine, or if you're good enough and you've got collaborators at the USDA who know what they're doing and can tell you how to do it, you can do an FMD vaccine. And the cost to develop that is simply the cost to demonstrate in research level that the vaccine itself will have some efficacious result. It is not the full licensing cost, which is astronomical. So, does this stuff work? I've heard this since the day we started. Well, does it work? Porcine circovirus 2, PCV2, one of the most widely used vaccines in the swine industry, uses exactly this production method, and it has for years. It's a baculovirus-produced subunit, produces the ORF2 protein, goes out the door, and it's used almost consistently right across the industry. And then we have our own development. We're developing, I saw wildlife on there. I've had a strong relationship with the deer farmers as a result of my work on CWD for years. EHD was ravaging the industry, bad in 2012. You know what? Subunits work for EHD. We're in the process of licensing an EHD vaccine using exactly this approach. We've shown very good serological responses in deer. We've got a good challenge model in mice that shows that it works. And we're going out to do the final demonstration of efficacy. Swine flu, I already mentioned, is on our target list. We know the target. Everybody knows the target. All you have to do is put it into this system. We have currently done this. We have just finished several efficacy trials. We're in the licensing pathway, and we assume imminent licensing, depending upon how long it takes us to get through our final process. These are strain-specific vaccines, which means that it's ideally suited to this kind of a platform, because as SIV changes or new strains become prevalent, we just produce a new construct that then responds to those. And finally, from a foreign animal disease, Rift, since this is an FAD. Rift is where we started. So Rift Valley fever, mosquito-borne illness, it's a virus, bunyavirus. Basically, you know, there's the target proteins. We produce those in vitro, we vaccinate against them. We know what protective titers are based on historical data, but more importantly, we can do challenge studies. And the short answer is it works. These are very simple. Control versus vaccinated animals. Protective titer of 1 to 40 is what the CDC and the Army uses as protective against challenge. We blow that out of the water, 120 and higher. Both mice and sheep do these titers. We have done research-grade trials in sheep, but we haven't done the full efficacy in sheep. So we have, however, done our full efficacy in mice because this is a BL3 containment. Control mice all die. Vaccinated mice just look at you and go on to happy, healthy lives until the end of the experiment. So it works. The most common criticism I've ever gotten. So how would this work for an FAD program? So going back to the characteristics that I think of when I think about an FAD program, there's all sorts of plans that have to be in place about how you're going to implement things, but from a tools perspective, PCR detection of the agent is going to be critical. There are field PCR machines out there, it's about the size of a suitcase. People have been working on them for years. They work pretty well. So you can get them out there. You need some way to detect serologically infection, and you need some way to vaccinate. So you have to have a vaccination target. You have to have what's called the DIVA target to differentiate them, and you've got to have the PCR primers. So when we make our stuff, we never actually use the infectious agent at all. We use the computer through PubMed or GenBank or colleagues telling us what the sequence should be, field isolates, whatever, in our database. We then use that to make our target production in our recombinant system. It's a recombinant protein-based system. That can then be tested for efficacy. We have a long-standing relationship with Kansas State and their BL3 facilities. We're currently doing work along with Plum Island on FMD, so they can do those studies for us. Once that is confirmed that shows efficacy, we then have a vaccine target protein that we can put in our antigen bank as a potential use for in the case of an outbreak. In addition, we can also develop DIVA reagents that we can use in our efficacy study to show that we have a way of differentiating the vaccinated and the infected animals, so another protein. So now we have 2 proteins. And finally, as part of making our construct that ultimately leads to our antigen, we develop PCR primers for detection of the infectious agent. We're very good at doing this target production and making the antigen, and we partner with people who will help us, or who are helping us to develop the assays. We partner with the D labs to develop the DIVA assays. We partner with Kansas and the ARS to do the efficacy challenges. We have other partners that can work on the PCR for us, both international and local. So the end result of all of this is that none of these end result deliverables have to go through full licensing pathway with the USDA. We're not developing an actual assay. We're developing a reagents that the D labs can use in order to run these tools. But it's not licensed, so that saves us money and time. We're not licensing these vaccines. The process is licensed. It's a fully regulated thing, but we don't have to go get the license for it. So production up to this stage for us, with the exception of the $500,000 efficacy trial, is not terribly expensive. And they're prepared. So if we have an antigen bank and if we have an outbreak, we can actually get vaccine out the door in less than a week, several days, as a matter of fact. And the antigen is concentrated, so it doesn't take much room. If we have to produce new stuff, but we have the construct, we can do that in just a little over 2 weeks and get it out the door. And if we have to produce a brand new construct, right now it takes us a few months, but our goal is to get that down to 4 weeks. So now you have an FAD preparedness program that is economically sound, that provides the reagents you need, and that doesn't overspend in the absence of a potential problem. So how much does it cost, or what does it do? Developmental cost. Licensed vaccine for a foreign animal disease. If you wanted to do all 23 strains of FMD, that's probably an underestimate, $35 million. Offshore production, what was originally in the ask was in the hundreds of millions of dollars to establish these relationships and have them provision available. Using this platform approach, we're looking at a little over $2 million. Is it DIVA compatible? The licensed vaccine, sure, we could insist that it be DIVA compatible. Right now they aren't, but they could be. Offshore production, probably not. Most of these are grow it and kill it, so they're not going to be compatible. Platform vaccines are, by their very nature, DIVA compatible. Can you come up with a companion assay? Maybe there will be for a licensed vaccine, maybe not. We have worked with another company who specializes in assays to develop an assay for DIVA for our Rift Valley fever, one that you would buy as a kit. They won't license it. There's no market. There's no money in it. Offshore production, highly unlikely there's going to be any kind of an assay. Ours? Yeah. That's a component. By the way, whenever I say ours, this is where my business partner will yell at me. Any platform will theoretically be able to do this so long as you have access to the proteins. So my push today is more to think about a platform. Obviously, I've got some self-interest in this on Medgene, but there are other platforms that either are in development or potentially could do this that would also potentially satisfy these characteristics. USDA licensed. Yes, the platforms are licensed. This is a licensed vaccine product, somewhere halfway between autogenous and fully licensed. Offshore production, sort of. Chances are they're not going to be inspected by CVB, so we're not going to know what's going on or what they actually have available. Strain specific, a licensed vaccine, could be. We've got the Ad5 vaccine for FMD right now, so if we get lucky and that's the one that hits, then we probably will be. If not, offshore production, maybe, maybe not. Ours, if it's not in the bank, like I said, in hopefully 4 weeks we could have it. And then efficacy demonstrations, licensed vaccine, you have to. That's the rule. Full challenge, everything. Offshore production, maybe. Could be basically like an autogenous where it's just been used, but we hope it works. Ours, that's part of our developmental platform. That's the most expensive part of the developmental platform as proposed, is the animal challenge with the agencies that know what they're doing in order to show that it works. So, how hard is this? Because subunits aren't going to work for everything. My board would like if they did, but they don't. So, just if we look at what's on there right now on the stockpile list, these are all targets for which subunit proteins are already demonstrated in the literature. Full challenge. You make the vaccine, you give it to an animal, you challenge it with something, and you see if it gets sick or not. Rift Valley fever, as we've already done. African horse sickness. VEE, equine encephalitis. Classical swine fever. The E2 protein, everybody knows that's what it is. Nipah, Hendra, avian influenza, Newcastle. All of these have been demonstrated efficacious as subunit approaches, and we know what the target proteins are. So, going back to the original plan, if we're looking at stamping out, development of those PCR primers will allow us to have rapid diagnostics available that we can potentially apply on-site with those suitcase devices if necessary. It's going to require an appropriate response plan, USD regulated, probably something the states and the feds are going to have to come up with. To be able to resume business, well, DIVA vaccination and surveillance. The DIVA assays, which will allow us to differentiate infected from vaccinated animals, will allow animal movement to resume. And that will be in coordination with the state labs and the people, and obviously the national labs that are regulating all of this. Eradication and returning to business norms. Ideally, an eradication vaccine with DIVA compatibility. Prescription-based would be that way. Maybe it'll be an eradication vaccine, maybe it won't, but the hope is to have things that are capable of that. You've got to have the diagnostic capability in order to monitor the eradication, to monitor the control of the disease. And probably there's going to be some degree of regionalization to control the disease outbreak and control the effects on the industry as a whole. So, to adequately address this, our little bit would be down here, and the relationships we've got with diagnostic lab and academics. But it's also going to require buy-in by federal and state authorities and by producers to recognize that this is an option that they want to actually go for, since ultimately the way this would have to be maintained long-term would be from producer input, producer support. So, with that, all of the work that we've done to date has been multi-institutional. Obviously, my university, South Dakota State University, and the D-Lab has been involved, K-State through the BRI, contract research work, as well as our collaborations with various other animal health companies. And then we work directly with producer groups, the deer farmers. We've had federal funding and support through USDA, NIH, and some of the others as well.